Hi, my name is Rachel Allen, and today I'll be presenting case connection number 4 for Biochem 1005. Today we'll be looking at two cases. The first case is ornithine carbamoyl transferase deficiency. In this case, looks at an infant with elevated levels of ammonia and orotic acid. Ammonia is a toxic byproduct of the amino acid metabolism. Since ammonia is toxic and not readily removed from the body by the kidneys, hepatocytes remove it from the blood by converting it to urea, a less toxic substance than ammonia. In addition, almost all the ammonia produced in the digestive tract is absorbed and metabolized in the liver to urea and glutamine. Urea is then secreted into the blood before being eliminated by the kidneys and the urine. Elevated ammonia levels in the blood can damage the central nervous system. Ammonia is produced by glutamate dehydrogenase, or GDH, which is also an enzyme that catalyzes the final reaction of the glutamine metabolic pathway. Ammonia production takes place in all of our cells in our body by metabolism of amino acids and other nitrogen-containing compounds. Ammonia is produced in our body by the process of transamination, followed by deamination. Transamination is the process by which amino groups are removed from amino acids and they're transferred to keto acids to generate the amino acid version of keto acid and keto acid version of amino acid. During transamination, an amino group is removed from an amino acid and transferred to a keto acid. Transaminases, or aminotransferases, are enzymes that catalyze these reactions. Deamination is the removal of an amino group from a molecule. Deaminases are the enzymes that catalyze this reaction. This process is very important in amino acid metabolism, as it is an important pathway for the anabolism and catabolism of amino acid. Transamination is important for redistribution of amino groups, the deamination of amino acids, the catabolism of amino acids, and the conversion of essential amino acids into non-essential amino acids. It also diverts excess amino acids towards production of non-essential amino acids and energy generation. There are two main mechanisms of amino acid transfer in the body. The first occurs in most tissues. The second mechanism is utilized by the muscles. In the first mechanism, ammonia is combined with glutamate to form glutamine. Glutamine is a non-essential and non-toxic nitrogen-carrying amino acid that is involved in detoxification and is the main storage and transport form of ammonia. It is also the main respiratory fuel of the intestine. During this process, ammonia is loaded into glutamate via glutamine synthetase to form glutamine, acting as a catalyst to metabolize ammonia. Then glutamine is transported in the blood, then to the liver where glutaminase cleaves it. Once cleaved, glutamate and free ammonia are produced. The second mechanism involves the transamination of pyruvate to form alanine in the muscles. Alanine is then released into the blood, transported to the liver, and converted to pyruvate through the process of transamination. This reaction leads to the formation of glutamate. Glutamate then undergoes oxidative deamination, catalyzed by glutamate dehydrogenase to form free ammonia. Due to the first pass metabolism, little ammonia from the gut reaches the systemic circulation. Ammonia occurs in an unprotonated form in the blood. Here again, glutamate is converted to glutamine by glutamine synthetase and is the only efficient way in which blood-borne ammonia can be neutralized through this. In astrocytes, glutamate is synthesized from ammonia and is transferred to neurons, which give rise to glutamate, which is an excitatory neurotransmitter. Glutamate released from neurons is transferred to astrocytes and is recycled back to glutamine. To summarize, glutamate dehydrogenase catalyzes the reversible interconversion of glutamate to alpha-ketoglutarate in ammonia. Ammonia is loaded onto glutamate by glutamine synthetase, and ammonia is unloaded off glutamine by glutaminase. In early fasting, gluconeogenesis increases as hepatic glycogen is depleted. The kidneys and liver Both provide glucose through gluconeogenesis, and if the fasting is prolonged, stored glycogen is depleted and glucose production produced by gluconeogenesis is released into circulation. In the fasted state, amino acids are converted into glucose to maintain glucose homeostasis. Nitrogen transport is connected to gluconeogenesis through the glucose-alanine pathway, where glutamate undergoes transamination reaction with a pyruvate to generate alanine. Alanine can then be transferred and shuttled through the bloodstream to the liver. Once in the liver, alanine undergoes an additional transamination reaction, where alanine interacts with the keto acid alpha-ketoglutarate, forming glutamate, and the remaining keto acid pyruvate. Pyruvate can then directly enter into the TCI cycle through gluconeogenesis, be used to produce glucose, which is then shuttled back to the muscle in our alanine glucose cycle. Nitrogen transport is exemplified by transamination reaction of amino acids, since amino acids contain at least one nitrogen atom. Through this reaction, the substrate for gluconeogenesis is formed in the fasted state. In this infant, the urea cycle enzyme that is most likely deficient is orthonide carbamoyl transferase, or OTC. And the least likely to be impaired is carbamoyl phosphatase synthase 1, or CPS1. This patient has markedly elevated orotic acid and low levels of plasma citrulline. This is caused by deficiency of OTC. Normally, this enzyme catalyzes the transfer of the carbamoyl portion of the carbamoyl phosphate to ornithine in order to produce citrulline. When this enzyme is lacking, carbamoyl phosphate accumulates in the mitochondria, leaking into the cytoplasm, and acts as substrate for carbamoyl phosphate synthetase 2, or CPS2, in the cytosol. This would lead to an increase in orotic acid, a normal intermediate in pyrimidine synthesis. As a result, the orotic acid in urine is elevated. Urinary orotic acid can also be increased in the deficiency of ornithine, arginosuccinate synthetase, arginosuccinate lyase, and arginase. In patients with OTC deficiency, plasma citrulline levels are often undetectable in acute phase. Based on laboratory results and clinical manifestation the infant has, the urea cycle enzyme that is most likely deficient is OTC, which is an X-linked genetic disorder and causes a mutation in one of the X chromosomes that can develop hyperammonemia. One urea cycle enzyme that is least likely to be impaired is carbamoyl phosphate synthase 1. When CPS1 is deficient, we would not have elevated orotic acid levels, so we know CPS1 is working and is least likely to get impaired. Subsequently, a deficiency in CPS1 would result in arginine deficiency. Therefore, arginine becomes an essential amino acid. In case 2, we'll be looking at 3 different scenarios. In scenario 1, we'll be looking at maple syrup urine disease. Maple syrup urine disease is a condition in which the body cannot break down certain proteins. The condition is named for the sweet odor of the urine in untreated babies. MSUD is considered an amino acid condition because people with MSUD have trouble breaking down certain amino acids, the building blocks of proteins. It is a rare, inborn error of metabolism defect resulting in decreased branched-chain keto acid dehydrogenase enzyme activity. The varieties of MSUD are caused by mutations or changes in the genes related to branched-chain alpha-keto acid dehydrogenase complex enzymes, or BCKDC. When those genes are defective, the BCKDC enzymes aren't produced or don't work properly. This enzyme is responsible for metabolism or breaking down branched-chain amino acids like leucine, isoleucine, and valine. These gene mutations are inherited on the chromosomes you received from your parents. Having MSUD means that you inherited one flawed gene for B, C, K, D, C from each parent. Deficiency of the branched chain alpha keto acid dehydrogenase leads to the accumulation not of deficiency of branched chain amino acids leucine, valine, and isoleucine. Leucine and isoleucine are considered essential amino acids. Since the screening test for branched chain amino acids came back positive, the infant is diagnosed with maple syrup urine disease. Treatment includes dietary restrictions for consumption of the branched chain amino acids, IV administration of amino acids that contain BCAAs, combined with glucose for extra calories, or formula fortified with LAT1 amino acid substrates, glutamine, alanine, zinc, selenium, and alpha-linoleic acid. The treatment will promote the utilization of existing leucine, isoleucine, and valine in the body and optimize the transport of non-BCAAs across the blood-brain barrier. Thiamine response, MSUD response to treatment with thiamine, or vitamin B1. Thiamine is a cofactor in the oxidative carboxylation of the keto acids formed by the transamination of the BCAAs. In scenario 2, we'll be looking at alkaptonuria. Alkaptonuria is an autosomal recessive disorder that results in the deficiency of homogencytate oxidase, or HGD, and the incomplete oxidation of two amino acids, tyrosine and phenylalanine. This enzyme is used to break down toxic substances called homogentic acid. Without adequate breakdown, increased levels of homogentic or melanic acid collect in connective tissues. As a result, the disease can cause onchronosis of the auricular cartilage, arthritis, blue-gray pigmentation of cartilage, face, ears, hands, and sclera, and daily excretion of gram quantities of homogentic acid, as well as cause severe arthropathy and cardiovascular valve disease. No amino acids become essential in AKU, and phenylalanine is considered an essential amino acid. However, dietary restriction of tyrosine and phenylalanine and tyrosine phenylalanine-free amino acid supplements lower tyrosinemia associated with alkaptonuria. Since phenylalanine is considered an essential amino acid, consumption cannot be completely eliminated. A deficiency in phenylalanine can cause tyrosine to become an essential amino acid. Absorbic acid increases synthesis of tyrosine, and an increased intake is also discouraged. Treatment for AKU is limited at this time, promising early reports of the use of low-dose nitonisone, an inhibitor of the enzyme 4-hydroxyphenylpyruvate dioxygenase, have effectively reduced urinary HC levels in patients with alkaptonuria and prompted ongoing trials of this therapeutic agent. Finally, in scenario 3, we will look at phenylketonuria. In PKU, an autosomal recessive disease and is an inborn error of phenylalanine metabolism that causes an increase in levels of phenylalanine in the blood. Phenylketonuria is a disorder caused by deficiency in the hepatic enzyme phenylalanine hydroxylase that converts the amino acid phenylalanine to the amino acid tyrosine. This pathogenic variant in the phenylalanine hydroxylase gene prevent metabolism of phenylalanine, and in this scenario, tyrosine can become the essential amino acid. Because phenylalanine cannot metabolize in the body, untreated PKU leads to a buildup of phenylalanine in the brain and causes central nervous system damage, including severe and irreversible intellectual disability, epilepsy, seizures, and acquired microcephaly. A dietary restriction of phenylalanine may be recommended, which avoids high-protein products like milk. This scenario, a low-protein diet would result in micronutrient deficiencies in the infant, especially vitamins. Dairy products like milk contain high amounts of B vitamins. Vitamin B6 and B12 are deficient in patients undergoing PKU diet. Vitamin B complex supplementation, including B6 and B12 cobalamin is needed. Vitamin B6 is significant to protein, fat, and carbohydrate metabolism, and cobalamin helps to produce and maintain myelin surrounding nerve cells, mental ability, and red blood cell formation. Thank you for listening to my presentation.